So without any further ado, welcome to the BD CellView Lens Plugin. I'm John Quinn. I am the Director of Science and Product Development here at BD Bioinformatics, aka FlowJo. My email address is john.flowjo@bd.com if you are interested in contacting me with questions or for more information afterwards. Okay, so always good to start a presentation with the why. So why are we interested in imaging? Why are we interested in using imaging? Focus of the discussion, of course, is going to be the software and the data analysis. But let's start with just even some of the background here on the why and the technology that develops the information that we're going to be able to use for analysis in the software. In any case, I was going to begin with, at the past CYTO meeting, I watched many presentations where somebody incredibly brilliant presented all kinds of complex math to ascertain and confirm some aspect of their measurements, of their flow cytometric measurements. Is this actually a single cell? Is the cell expressed in the membrane or not? How closely are various expressions packed? Are they on two different cells, etc.? And time again, right after I was done being amazed that somebody was able to work out the details of some kind of algorithm or technique or approach, it occurred to me that they could have just looked at a picture and it would have been really simple and easy. And so I'm pleased to show you technology today that will allow for an easier path to QCing, for figuring out the cell's morphology or the spatial resolution, and to do so in a high throughput system that is capable of measuring thousands of cells per second and then sorting live cells as well. So where does the data come from? The data, and I'm going to give you just a few slides here at a very high level. There will be many other presentations in the world done by somebody who is central to the technology's creation. But for now, it's probably enough to say that the BD FACSDiscover recapitulates two-dimensional information from measurements in one direction by essentially dividing a laser beam into many small beamlets. I've heard that term used and I kind of like it, so I'm going to go with it. That are each at unique frequencies that allow for tracking information from each beamlet, so different frequency, different beamlet, and then assembling all of those images from a beamlet, all the information from a beamlet into a composite picture. So no cameras are involved here, which allows for much greater speed since there's no limitations based on shutter speed or any other aspect of photography. And then additionally, once we've got these generated images, we can make a variety of measurements on them and include a bunch of image-derived parameters within the data. I want to show you this movie that BD Marketing put together that I think is 30 seconds of just coolness that shows the laser beam being split up into all these little beamlets, a cell passing through them. So we're collecting information through each one of those beamlets. Each one of these is a waveform that then we, like this image, collect, turn sideways, and put back together into the image of the cell that we are then going to present to you for viewing within the CellView plugin. So there you go. I think that's just nice work there by BD's marketing team. Good job. That's beautiful stuff. Okay, so that's where the data is coming from. I mentioned that we are also going to provide a bunch of different image-derived parameters. So in addition to traditional fluorescent parameters, which we of course collect as well, and the random images, which I just showed you the movie on, there's going to be 6 image detectors that we can derive information from. And so you can get light loss, forward and side scatter, and 3 different fluorescence channels that you can append to the target of your choice. And for each of those 6 image-based parameters, the FACSDiscover measures 7 different derived parameters, 7 different aspects of that visual parameter. And so last set of slides before we talk about data analysis is just I want to make sure that we're level set and we know what each of these different terms means. And so first, eccentricity. And you can see on these slides that we have some graphic over here showing the difference going from a low eccentricity value to a high eccentricity value. So eccentricity is essentially the roundness versus oblongness of a particular cell. The graph over here on the left imagines that we showed eccentricity as a histogram and gated 3 different regions. And you can see there's a color code, and this will be consistent for all the rest of these parameters, a color code showing low eccentricity what cells look like if they fall into this end of the histogram, so this more teal color, and you can see a roundness to the signal that it's measured. So again, each of these measures come from, we can get them off of any or all of the 6 channels. So you could be looking at the roundness of forward scatter or the fluorescent parameter attached to channel 1, et cetera. We have them all, we record them all, so this is a very information-rich data set. Medium eccentricity, and then you'll see what this high-end eccentricity looks like. It's very oblong in terms of the, where the green signal's being collected. And so you can look at these pictures and think, well, eccentricity, for example, might be a really nice way to identify doublets. Gives you a picture of the morphology, depending on maybe it's one of the scatter or the light loss parameters or something you're looking at and so forth. We can do just the max intensity. What is the brightest portion of the signal that you're looking at? And so you can see again on this, this color-coded histogram, so dim to middling to very bright fluorescence. Is there perhaps a particular target, a fluorescent target that you were looking to find the cells that are brightest for? Well, then perhaps the max intensity might be of interest to you. We can measure the size. So just you can see the difference here in terms of growing measure of fluorescence. Could be related to the cell size, could just be related to the amount of something else you're measuring on the cell, I don't know, DNA content or something like that. So maybe this is a nice identifier of cell cycle. And this one's pretty straightforward, where smaller things are here and bigger things are there by breaking down the histogram. We can look at the radial moment. So essentially, from a center point, how much does the signal that we're detecting for a particular parameter expand outward? So from very tightly grouped together in the middle to all the way at the outside of the cell. And then again, you can see low and high radial moment pictures right there. Correlation. So are 2 different parameters expressed in the same place in a target cell? And so here's uncorrelated, where we see a lot of separate orange, a lot of separate green, etc. And then over here, correlated, where those colors are overlapping, whatever 2 things we're measuring here are co-expressed in the same place, and we have a lot of yellow. All right, delta center of mass. So you can pick 2 channels and ask how far apart they are. How far apart is the centroid? And so you can see the purple and the green here, depending if they're very close together versus middling versus far apart. So that's a pretty interesting set of graphics. So I really like how this worked out, in that we've got some high delta center of mass where we can see 2 pretty distinct types of cells that just happen to be caught in the same image. And then the middle ground, that middle ground could potentially be the sweet spot for some kind of cell-to-cell interactions, where we see 2 different types of cells that actually appear to be in contact with each other, as opposed to perhaps the same cell expressing both things. Diffusivity is essentially a measure of, is the signal more punctate or more consistent? Do you see, those individual dots or not? And so again, another way that we could look at in this histogram, some low diffusivity, medium. So here's the punctate showing up again to high, just sort of a overall expression of the parameter is listed there. Okay, so those are the derived parameters. And so what is our workflow? How do we get this data? So the data are collected on the BD FACSDiscover software. That is going to export both the FCS file, and then for each FCS file, it will create a CVW, a waveform file, that can be extracted then into your images. And so for a 10,000 event file, here is some representative sizes of those things. You can see the images are going to be fairly intense. So this is probably the moment I could mention the BD Research Cloud and say that might be a good place to store it. It is also a longer term plan to park the technology involved in the image extractor in the BD Research Cloud, so you can upload just the CVW to that and then open it into FlowJo. So FlowJo, data analysis software, of course. What's maybe not obvious is that then all of the tools in FlowJo are then accessible to use on this data. So that if you want to do gates, or if you want to do graphs or tables, et cetera, but also algorithmic analysis or t-SNE, et cetera, it's all there. It's already built in and you can use it. The tool that we are talking about mostly today is the BD CellView Lens Plugin, which gives you the capacity to link together the images and the collected FCS files I'm also going to give a few moments to Hyperfinder, which is another plugin that when combined with the BD CellView Lens allows for the completion of the computational sorting workflow, where perhaps using tools in FlowJo, I don't know, cluster or something like that, or train something to identify cells that are of interest, we could ask Hyperfinder to create populations using sort capacity gates and open it right back up in Chorus where we can go back and verify anything that we've identified computationally. So that is our overall workflow that we're going to talk through. Okay, step one, how do I get the BD CellView Lens plugin? So it is a plugin available on the FlowJo exchange. So flowjo.com/exchange. There it is. That'll get you there. There are a bunch of cards in the exchange. The one that you want is the BD CellView DM Lens. Just hit the download and it appears on your computer a moment later. The only step for this plugin to use all the basic functionality is just to put it somewhere on your computer. So drop it in a folder of your choice. And then from the FlowJo preferences, and it's the diagnostic preference specifically, you'll check scan for plugins and tell us where it was that you put it. Anything in this folder gets added to the plugins drop-down menu when you start FlowJo ever after. So one restart would be needed at some point for it to appear in the menu, and then you are ready to run. Okay, so then now's workflow is to load a file. So I've loaded my .fcs file that has some associated images. I go to the plugins drop-down in the workspace band. The first time that you want to look at some images, FlowJo will ask where they are. It's going to assume that they are in the same place as the .fcs folder, and that there's a particular file structure because the FACSDiscover will start putting your images in folders, named in a particular way, a bunch of zeros, etc. The more images you get, the more folders you get, and so forth. Generally, you'll just have to say OK at that point, and then the window pops up, and you've got images. So then how would you like to use those images in association with the FCS files? This is the part of the workflow that we now mention as all optional. There are a lot of ways you can go. How you want to proceed is completely up to you. So step one, I might suggest, there is some utility, of course, in adding whatever kind of manual gates you would like to do. I would like to see cells that I think by traditional methods maybe are single cells and start from there. Or maybe I would like to make gates that also combine imaging parameters. So you can see in this just example gating scheme that I've thrown on there, we're using something called ERM singlets. That's my eccentricity and radial moment singlets. So I can just mix in any of those derived parameters into a gating scheme. Where the CellView Lens, I think, adds to your workflow really is that then you can adjust gates based on imaging parameters. So I could do something like create a CellView Lens plugin on any of a couple of populations. So I gated what I called poor and good cells by side scatter there, and then use the CellView Lens plugin to make it so, to make those designations true. Incidentally, you can start the plugin on any one node. And if you just drag and drop the CellView Lens node to another population, it will create a CellView Lens window for just those cells specifically, but maintain whatever filter and preference settings you've applied so that you can consistently look at the cells. So here, when I say, how do I adjust the gates to make it so, I can click a button and my graph window comes back up. And as I move the cursor around, I can see cells that look better or worse. And I can identify if there is a portion of my data that seems to have a lot of things I want or a lot of things I don't want. In this case, I was just looking for two cells together and moving the gate. So they ended up in the poor gate, one cell at a time in the good gate. But alternately, I could do something like look for some rare population by trying to make sure that I've put cells that express, one of these fluorescent probes. As you can see, actually, I happen to have found a cell that's got some pink on it, which I think I was associating with the PDCs in this experiment. Okay, and then we can look in the image wall at more than one cell at a time and see how we've done. And this is all live updates. You can adjust the gate and see how the image wall updates as you're going. So more so than just moving through one cell, but also fine. You can also look at a large number of cells. So here's the poor. We can see that quite a few visible two cells together in these plots. We can see the good where we've got generally one cell at a time in those plots. And then one thing I want to mention is that you have the ability to sort these images based on any of the parameters that we've measured, image-derived parameters included. And so I've sorted on eccentricity. So I know right away that I'm getting apples to apples comparisons of these two populations, and that perhaps I'm putting them in the order of best to worst or worst to best, depending on whether I sort ascending or descending. And it just makes it a nice way to figure out if there is a parameter you're gating on, that you're ordering them in terms of that parameter. So if you adjust the top of the gate or the bottom of the gate or something, you're trimming off the edges and moving more towards the population that you are most interested in. And all updates live again. So this is relatively straightforward to go through and say, those are the cells. Okay, so other options. That was a heavily manual option that we might want to use for analyzing the data. But again, all of the tools that are available in FlowJo are available here too. I realized when I went to full screen, I minimized the chat. Okay. There might be somebody having problems with the audio. Overall, it seems like it might be okay though. So we will hope for the person that was having some audio trouble, turning up the volume, tightening up the headphone connections did the trick. And if not, we are recording this and it will be posted to the FlowJo webinars website shortly after completion. Okay. Moving back over here. Okay. Moving back over here, we can use clustering, dimensionality reduction, any of the algorithmic analysis tools that we want to. And I think really important to mention is that you can use those tools using fluorescent parameters, image-derived parameters, or a combination thereof. So in this example here, I just went to one of my midstream populations, and I said I would like to cluster these, and I would like to do so using my fluorescent parameters, but also all my image-derived parameters in which I've attached CD123 and CD141 through as the image parameters, and I've done it with max intensity, eccentricity, radial moment, all of those imaging parameters included in both my clustering and my t-SNE results. So t-SNE up there. And so, we get what ends up looking like a clustering result that lines up very nicely with the t-SNE result. It's always nice when you feed all of the parameters into two different approaches and they give you results that, that look like they match up. t-SNE put together groups of cells that it thinks are similar, and clustering without knowledge of what t-SNE is doing, clustered them and put the similar cells together as well. And then worth mentioning that all the CellView plugin tools work here as well. So I could at that point then go and pull up a graph window that is going to allow me to mouse over and see if any of these populations are expressing either my pink or my green image colors, indicating that they've got some of the fluorescent parameters I was looking to measure and identify the particular sets of dendritic cells I was looking for in them just through mouse over. And you will tend to see if clustering or dimensionality reduction worked fairly well, that similar cells have gone together. And again, using the imaging parameters, we can really, enhance what we're doing in terms of identifying interesting populations of cells. Okay, Hyperfinder. So, cool. I've done that. I've used t-SNE. I've used clustering. I've identified a population. Perhaps it's something like FlowSOM cluster 1. How do I go in and find that? Or alternately, maybe I've gone in with manual gates and identified some kind of population, etc. And I've done so using a ton of parameters, and maybe there's even a more succinct way to do it. So either one of those cases is useful as a way to get Hyperfinder to make a pretty streamlined gating strategy to get down to a population of interest. So you can see I've started the Hyperfinder window here. On the left, I have picked a variety of fluorescent and image-derived parameters, going for, there's a relatively long list of parameters here. What's in view is the fluorescence, and I've also, clearly got the light loss, the side scatter, etc. images here. But if I can, scroll, if I was able to scroll down on the screen, you would see that I was grabbing the CD123s for light loss, for eccentricity, radial moment, etc., 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 so that I can go through and feed all that information which the cytometer can measure, so it can use for sorting, into the algorithm to do some gating. I see an issue in the chat of, is there any issue when you use fluorescent parameters plus morphometric parameters doing dimensionality reduction in the same time? Is it the same scale? That isn't Awesome question. The answer is no, it's not the same scale, but FlowJo is going to use actually what you see visible. It's going to use like the screen resolution. And so it's a nice idea if you look at your scaling, and I do actually put a slide in that says just this, look at your scaling beforehand and trim out the white space, make sure that you've got reasonable display for the range of data. I'll talk more about that in a minute, but great question. So back to Hyperfinder. So yeah, we put these all together and we can go in, run a Hyperfinder. It does its thing where it goes in and says, okay, I'm going to take some of the data from this sample, the target population that you're looking for. I'm going to take some other bit of the target population and use it for, I'm going to take some of it and use it for training, some of it and use it for validation. And I'm going to start making gates on any combination of parameters that I think is going to get the job done. And then once it finishes, it spits those gates out as high-five or Hyperfinder and gives you those gates within your workspace. So starting, you always will start from the top level in Hyperfinder. It pops them there and it says, here's the gates that I think get you to the population you're looking at with some listed F measures. So the harmonic mean of accuracy and precision. And then, exciting, if you go back to Chorus, there's just a FlowJo button in the upper right-hand corner. If you press it, you can simply import your FlowJo workspace without any other kind of intermittent step. And it brings those gates in. It brings in all of your gates, including the Hyperfinder gates. And at that point, you can simply sort. So I got to learn how to use the machine out there. And it was kind of amazing to me when the data set that I'm showing you here, we just went back in and started getting some plasmatoid dendritic cells popping into tubes just directly from our FlowJo workspace that we imported back in. So really cool, really cool. Okay, other things that you could do as part of your workflow to help ameliorate the images, your ability to resolve them and so forth. There is a tool for just going in and thresholding all of your images, however you want, hiding or showing different parameters. And there's this tool up here where you can adjust the threshold for what is being displayed. And I'm generally looking at this preview window of the overviews as a guide for how to do it. My general approach there is to adjust your thresholds, your gamma, your min-mass, etc., so that you see the signal and you cut out the background. And you can do that for each of the channels individually, look at how they appear on the overlay. You can turn off certain channels or not. I found particularly the light loss channel is nice for seeing, the edges and basically physical boundaries of the cell, but it brings in, it's very bright, brings in a lot of light into the background. So you get more of a gray background than this. I like the stark black if I turn off light loss. And I found that the side camera imaging parameter does a pretty nice job of actually showing you the basic shape and size of your cell. Up to you, forward scatter could be used as well. So this is just showing you a choice that I made here to really highlight a handful of functional markers and just contrast them with side scatter to get a feel for how big and where the cell was on the screen. There's a variety of different types of overlays. You can pick blended, transparent, or solid. Big fan of the blended myself. And then once you've done this work and said, that is what I like, you can either import or export filters. That means you can just save a filter set. You can export it so that anybody else could use it as well. So that if you and a fellow researcher happen to be looking at similar data sets, and one of you happens on a nice way to view the data and you'd like to make it consistent between people's work, have that, feel free. Hit export, and then anybody else can import it into their workspace. Okay, so some recommendations. Interesting. Good question on can we import filter sets in FlowJo images settings from Chorus? You cannot at the moment. I had not thought of that. There are very similar settings in Chorus, they're not identical, but that is I'm writing that one down as a good suggestion of data that we should maybe be able to pass between different BD software. Great call. Thank you, T-Bolt. Okay, some recommendations. In FlowJo, we will identify what cytometer any data came from using the $CYT or cytometer keyword. If you go to the preferences and cytometers, you can see all the cytometers that you've added data into your workspace at any point in the past, or any workspace, I should say. If you pick the cytometer that you're interested in looking at, So interestingly, it always just shows the top cytometer highlighted when you enter the preferences. It is applying the preferences based on the CYT keywords. So if you open it up, you'll scroll down, pick the cytometer that you're interested in, and that will apply the preferences like that. So not to be concerned if you come in and it's saying you're using Diva, it's actually just a list of preferences. I think importantly for the FACSDiscover, there are a ton of parameters that get collected. I think we're fairly familiar with there being a -A, a -H, and a -W, the area, width, or height of the electronic pulse that is triggered in the PMT or whatever collection device you're using as a cell passes through the electronics. And you also get a -T with anything that comes off the FACSDiscover. So you've got 4 measures of every single parameter in there. You can see in my picture here, and I will blow that up, that these parameter filtering options down here, I've chose to filter out the -Ws, the -Hs, and the -Ts. So that reduces the list of parameters by 75% right off the bat. And that makes this much more manageable. With 4 versions of every parameter listed, it can be hard to scroll or find the one you want. I highly encourage using the search function as well to get through larger lists. But then importantly, there's also these Boolean linkages for how to use these parameter filters. And this one's cut off with ... but it's and not. So maybe you want some of the -Hs or -Ts or -Ws. I will leave the discovery on which ones are most useful to you, but I will suggest that having the height version of the scatters tends to be pretty useful. So I say, and if it says, either FSC or SSC has the same root, if it's got an SC-H in it, please keep that one. I made that an or not. You can see from the scroll bar that there's a few other things that I thought, well, maybe I'd rather not exclude that. So up to you, but I really recommend parsing down your parameter list a little bit so that it becomes a bit more manageable. And then, as I believe it was T-Balt brought up earlier, scaling. So it's really important if you're going to do any kind of, well, I mean, even just to see the parameters nicely, it's good to have the scales right, but it's especially important if you're going to do any kind of clustering or dimensionality reduction to make sure that the parameters you intend to include are on reasonable scales. And when I say reasonable, I mean, they're not all smashed onto an axis, the data themselves are visible. So nothing, nothing too crazy. You just want to make sure they're visible because we actually do use the data space. The visible space that you see essentially gets binned up. And what we're looking to do with those algorithms in almost all cases is to find things that are either similar or different and push them together or push them apart. And so you want to say, if you've got, in this little simple example here of 2 sort of peaks, a smaller one and a big one, et cetera, the things that are all in this big peak you'd like to see go together in clustering or dimensionality reduction, things in the little one maybe get together. And you're letting the algorithm know that these are considered fairly different because you push them all the way to the edges of your data space and didn't leave a whole bunch of white space. That if we did have white space and we smashed all of the info into a very small bit of our data range, then you would be telling the algorithms that they're all pretty similar. So it's nice to go in and look through your parameters. You can multi-select. Many of the parameters are going to have common ranges, like all your fluorescence, all your max intensities, etc. I do have just the cursor I'm using on my scene, Cecilia. I'm hoping to describe most of what I'm talking about in words, and I don't think I've used the cursor very much. So that might be why you're not seeing a lot of pointer. At this point, I'm just talking about the right side of the screen. So anyway, that is scaling. It's worth doing, it's worth looking at. And I do understand that there are now a lot of parameters, and because all of the image derived parameters do have very variable scales compared to your fluorescent parameters, will all say be on the same scale, more or less, that we do need to add some tools that makes this easier. So it just, a bit of active research right now is an auto scaling parameter or an auto scaling tool that we might first give out as a plugin, and then we'll attempt to build in directly into FlowJo in the near future, so that you can click a button or give it a set of rules to auto apply a set of scales to your data so that you don't have to worry about, maybe trying to find 20 different parameters and adjusting their scales. But for today, important to look through and spend a moment or two just making sure your data are all on scale for all the variety of parameters that are here. Okay, and then while I'm talking about evolving work, there's one more tool that we are building It is in right now, though it requires some special configuration and adding some libraries and only works on Macs at the moment. So it is still a work in progress at this point. And we also have to characterize it and test it. But I think something that's going to be really important for the future is the ability to use artificial intelligence, to do like the cell identification. So the workflow that I pointed out was that we could pull up some graphs, we could mouse over it, we could identify interesting cells, we could pull up a wall of cells, and we could try to use some gates to isolate the cells we want from the cells that we don't want, etc. Probably the better long-term plan is to maybe do that once, maybe to find a small population that's exactly what we want, and then ask a neural network to learn what those images that we like look like compared to images that we don't like, and then just go in and find other images that look like the ones we want and go pick them out. So this is just showing that with a little bit of configuration, you would see this gearhead icon pop up in the bottom right-hand corner. If you're interested in doing this and currently on a Mac, I will just say contact me, and I can give you instructions on how to make that hidden preference appear. Now, again, still working on this one. But anyway, you can then have it reference a bunch of neural network models that, again, try to learn it. What's being shown here is you get some plots of how well training versus validation were classified, how accurate it's going to be, and then apply that to populations or other samples, for example, that you'd say, okay, I spent some time telling you, machine, exactly what images are the most appealing to me. Please find them. Please find the ones that have a lot of green fluorescence or pink fluorescence or whatever it might be, or a shape that looks like this, or one cell as opposed to two in a plot. So that is our future direction. And then the workflow that I imagine, sorry, too many arrows on the slide actually. The workflow that I imagine then is, you'll import your data, you'll put a few simple gating strategies, you can compare gating strategies, you can use it to identify some kind of population that you're interested in. You can use the image processing tools to make the populations of interest really stand out, or the colors that are most indicative of the cells you want, really shine. Have a neural network, learn what those look like, have the neural network then, go back and classify, future data sets, or even the larger population of the data set you're working on to find more of the things that match exactly what you want. And so we can really take advantage of the high throughput aspect of this particular technology. Okay, that is the slide portion of this. Just checking the chat. Looks like we're doing okay in terms of questions. Let's get out of slides. Okay, escape button, do your thing. There we go. Okay, I have a couple versions of this workspace up here. I've got one that's just at the start and one that's probably done. And we can bounce back and forth as needed if we come to anything that might take a little bit of time. But I figured I'd start right at the beginning to talk through all the different things that I showed, or at least most of the different things that I showed in the in the slides. Okay, so first off, preferences were just the heart, diagnostics, and this is the spot where you say scan for plugins. You can put your plugins anywhere. You can see I just stuck mine in the applications folder. So applications, put another a folder called plugins in it, and it just reads any plugin that's in there and puts it in this list here. So that when I pick a population, I could start here, for example, and go plugins and BD CellView Lens, it's just pulling up the plugin from that folder and showing me the images. And I'm sorry, I guess it was smart enough to remember that I had done this before to complete the workspace and remembered where the images were when I made a simpler version of this workspace that did not have any work done. So it skipped the spot that said, tell me where the files are. But I will mention, if you ever want to reconnect them, or it doesn't, I don't know, whatever, or you need to redo it, there is in the tools open event to image file mapping dialog. So this is the dialog that would have popped up that said, hey, I think based on where the FCS file is, this is where I think your images are. If not, hit set folder. The rest of this is basically pretty automatic. And I will just show you, I just have this stuff sitting on my desktop at the moment in a file structure that looks like this. So stuck an image filter, XML file that I might import later in, images right here, FCS file there, dragged and dropped the FCS file in, here's the images. Okay. There's a question. For what I understand, you need to set the image settings, brightness, blah, 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 prior to recording the Discover S8. FlowJo probably has to play with those parameters afterwards, but the starting images will have the settings set in course. Stefano, yes. There are, this is, I don't have the course software handy right here, but yeah, you're exactly right. You've got a very similar set of tools in course that you can set the images up. And then right in FlowJo afterwards, you can tune them if you would like. And if you wanted to tune them, good segue, there's this little tuning button right here where you can go in, you can include certain parameters or hide them from your overlay just by clicking on or off. You can associate different colors with specific parameters that you're interested in. Here's what it looks like if we include the light loss. I don't like how the background goes so light, so I tend to leave that one out. And then you've got your, for whatever parameter is that you're working on. And let's pick one that actually has a signal here. At the moment, I'm clicked on a cell that does not have any 123 or 141. You can choose to adjust your thresholds, and you'll see how that impacts the background and so forth. Your gamma, which is essentially like a sigmoid-shaped threshold for how much signal you want to let through compared to background and so forth. Blended overlays versus solid. Actually, I should click on a different cell that actually has some color in it. So we'll at least get one here that's got some green. Okay, so we can see some of the different, some of this in work. Oh, there we go. That is too low on the gamma. A lot of background being introduced. Anyway, so it's sliders. You can check the overlay picture and see how it looks. That was probably too much again of the green. We'll go with that. Anyway, a little too much background maybe from the scatters as well. Anyway, closer to what I had there originally. Anyway, so over here is the various sets. You can go default, auto color smooth. I'd be 11 color in there. If you want to import or export, it's just the button here where we would say import filter set. And yes, since I actually went through the work once, it still has it imported there, but it would just be a matter of going to desktop, CellView webinar, pick the 11 color. Okay, so there is image filtering tools built in there. If I want to see where any of these images are on a FlowJo plot, it is the button here. It turns yellow around the borders. And then once you move the mouse in there and it knows you're ready to look at it, it turns green. And then you can just mouse around and see what cells look like in different spots. So right down there, a lot of singlets in my singlets gate. And then for fun, I made a small gate down here that I called bad. And we can see that that's got an awful lot of doublets in there. Okay, we can similarly click on images in the image wall. I should put these next to each other and have the cross appear where it is. So, okay, looks like my doublets happen to be right on the bottom edge of that singlets gate. I mentioned that sorting is a useful thing. So you can sort by any of the parameters that you want. There's search bars in here. Take care of these, take care to use those. Eccentricity was a pretty good tool for showing things. And we can also do more rows. We can do less rows. We can zoom in and so forth. Let us sort the other way and find the bad cells. So cells where there's, lots of doublets and see where those tend to appear. So it looks like we've done, a reasonably good job. That could be an interesting one. So there's definitely certainly some assays in which, probably not, probably just two things that happen to be stuck in there together. Maybe that's the interesting one. We've got a few different cells and all of them look like some kind of dendritic cell and all of them in close proximity. So we've got some interaction. Anyway, there's definitely going to be some assays where perhaps you are actually looking for the doublets. And so maybe that's a tool that could be of interest to you. Okay, at any point you can take the camera button to take your current image wall and add it to the layout editor. And it'll just be everything that's in here. And so maybe we want to go back in and see where 773 was and see if we could exclude that. Oh, we have excluded that. Good. Okay, cool. From my actual singlets gate once we would start using that. Question, doesn't side scatter look better when data is taken at longer wavelengths where the transmittance to cells increase? For example, can data be attained at 730 nanometers and 780 nanometers? Transmittance of cells increases from 750 nanometers. Kanye, that sounds reasonable, but I am not your instrument person. I will pass that along to anybody who makes an instrument and is on this call to think about that. Okay. I think that is, oh yeah, I guess we could switch to channel view if you want. So we can see all the different channels, but I do kind of like the wall view where you see one view, your selected one, whichever one it was that you selected last over here. So if I just want to see, I don't know, side scatter view or something like that, we would just see the side scatter of all of these. But I do like the overlay. When now we're looking at the overlays of all of them. Okay, so there is your basic tools within the CellView Lens plugin. See a raised hand. Da-da. Okay, we have a question. I understand the images are not RAWs, but in fact preset from chorus. The filter settings just to adjust better afterwards. It needs, so probably no need to adjust after recording. Everything needs to be settled online, I think. We have certain defaults that we will use when we show the images. We've got another set of defaults that tries to match chorus pretty precisely. And we'll definitely work together to make sure that we can pass these back and forth. You are getting the FlowJo default, so you may want to actually come up with your own filter settings that make them look as similar or as dissimilar as you prefer them to be. All right. In here, I did want to show them that you can create a CellView wall just by dragging and dropping to a particular population. So if I want to look at my good ones, hopefully this is a more refined set of cells where now we've got mostly singlets. If I wanted to go downstream and look at relatively rare populations to see if I did a decent job of identifying them, I can do so. So we can double click in here and see if we have a bunch of pink or green for these two populations. Okay, we definitely did good with the PDCs. Okay, so ways to go and use this tool. So this is a nice validation. Does my gating scheme work? If I'd wanted to do anything like clustering or on t-SNE, etc., it was just a matter of picking the population you're interested in and using the tools as normal in FlowJo. So you go up to workspace and click t-SNE, or plugins if you're more of a UMAP person to come down here and UMAP it or cluster it. The images that I used in the slides, I just pulled directly from this workspace, or at least a similar workspace that I have opened here. So we can shortcut the trouble of actually running t-SNE or UMAP just to say, you will see if you run any of these derived parameters appearing. I ran them on the CD19, CD3 population that you can look at in the CellView lens. And then if you go into the viewer, you'll have the ability to choose the t-SNE parameters as the ones that you would like to look at and to see which of these populations perhaps have the most interesting. Oh, it was that little guy over there. That little one looks like that's my plasmatoid dendritic cells. Okay, should have shown that in the slide. Whoops, I think I grabbed this little population up here because it seemed to have a lot of the green, which might have been my MDCs. Okay, so just to show that there is a nice harmony between the way that our tools work together. Okay, and then in this tool, let's actually go in and grab. Let's grab which one of those populations was the PDCs. I'm going to go to my layout. I'm going to say it was the pink one, which is pop 5. Okay, so we're like, hey, pop 5, based on a ton of parameters that I put into my clustering algorithm, I think that's the one that I would like to make sortable. So if I pick top level population, I can go to workspace, plugins, and hyperfinder. Did select a population. Okay, there we go. And I could choose all of the parameters that I'm interested in allowing to be used for sort. So maybe I do want some of the side scatter, the light loss seems like a useful thing. There's 2 different light loss measures. All the fluorescent parameters perhaps should be fair game for producing a gating scheme. We could go down here and grab, too far. We could grab our eccentricities and so forth of anything perhaps that was one 41 and 123. So center mass, why not? Some long axes, let's go definitely max intensity. Radial moment, total intensity, all seem like good ones. Okay, there's the eccentricity. So you pick all the things that we're interested in. You've got a variety of different gate types you want. Polytope might, theoretically might be the best. I kind of like the polygons. That's totally your call. What do you think is going to be the best? And then what pop do we want to recapitulate? Well, pop 5 was my PDCs. Let's say that. So we say, okay, Hyperfinder again is going to take some of that target population, use it for training, take another portion of it and use it for validation. It'll give us some graphs to show us what the gates it produces look like. Okay. So it is making a series of gates. I probably could have taken long axis moment and cut off a little white space on that one, as it does look like it's made a lot of use of long axis moment. Who knew? There we go. Okay, so F measure is now refining to make sure, the Hyperfinder is now refining based on the F measure as it adjusts those gates a little bit this way and that. You can see the overall F measure improving as it makes iterative improvements to the gates. And in the end, it's going to spit out a bunch of hi-fi gates that we can then just open back up in Chorus if we wanted to sort on something that was simply identified as cluster 5 using FlowSong. Let me go see what's in the Q&A. It looks like there's one question there. Let's see. The main challenge of the system is that a vast majority of our users only do a few colors and don't have experiments that are designed to take advantage of the technology. How would this technology be useful for mundane, older studies established in something like the Ecury, rather than cutting edge stuff that's only been requested by one user so far? I expect it may be used for troubleshooting experiments where populations don't separate well on scatter plots or unclear, etc. Yeah, sure. Daniel, I will say it's probably not the optimal use of a super awesome high dimensional machine, but any experiment I think can be improved based on using the ability to visually just QC the data at the very least. I think if you've seen a talk where somebody presents imaging at any conference or user group meeting, it feels like it's been really easy for the presenter to walk up and go, here's your typical singlets gate. And look at that. From in that singlets gate, I can find a whole bunch of garbage, debris, et cetera, that we've all have just used the law of large numbers to assume that, well, most of it's going to be fine. So you can definitely get cleaner data no matter what. And then if you have an experiment that even is in very few colors, but perhaps relies on something where more, maybe not morphology, but the spatial resolution is super important. Like, are these two cells together expressing, maybe one parameter each that tells me what kind of cell it is, and are they in close proximity, maybe indicating that they are doing some kind of cell-to-cell interaction. That's the type of experiment where this tool will be really useful. I think it's definitely experiment-based, probably not useful for every experiment, et cetera. And, definitely you probably wouldn't want to just run mundane experiments on the S8. Would be my assumption, unless having really clean, pure results was very, very important for the target of the experiment. All right, that was in the Q&A, and the Hyperfinder is done. All right, so we've got our populations. These are these hi-fi gates right here. It's telling you what parameters they gated on. File, save, and you're ready to open it back up in Chorus. And that would be that for getting a very refined population back. Okay, let's just check the chat, and then I want to show you, cool. Yeah, I want to show you just that. If we pick some kind of upstream population, yeah, I think that was fine. We could do that since I've already put the CellView lens on it. Just a preview of what we're looking at in terms of trying to add AI to this. I'm going to click the little smart AI guy button. That brings up this interface here. So the things that we're trying to do. So, oops, sorry, sent it to the background. We can add a variety of neural networks. So neural networks are essentially some kind of algorithm that takes the measures you feed to it as inputs, and it passes them basically through a series of weights and says, all right, if we pass the series of weights and we get one number, you're this type of cell, or this, this class of data. And if you pass through the weights and you get another one, then you're a different kind. So we're generally trying to separate two things from each other. So I might go back into this workspace, and actually I've probably done it already. If I wanted to sort PDCs and use that as my example of here's PDCs to train a network, I just made a NOT gate. So just from tools, Boolean, make a NOT gate. So these are not PDCs. Everything that was in the CD14 negative population that is not a PDC appears here. You know, perhaps I could have done more and done a NOT gate or something up here so that I included more examples of not PDCs that weren't quite so exactly close to the PDCs that they've matched all the parameters upstream. I'll let you formulate the exact use case that you want to go through. But you're basically trying to find two populations. Now, where do I find my window that I'm working on? To say, I want these and not these other things. So you've got a variety of networks you can choose from. It's relatively easy once you've configured this to start up and talk with Python to grab any kind of existing neural network model that's anybody's written for Python and have it appear on your list. But we could pick one of these functions. And you could say, well, give me two things that I'm interested in discriminating. So I might pick my PDCs and my not PDCs. Okay, the neural network code is right there. It's not a lot. Python has just a ton of built-in, functionality that you can just call. You can say, okay, that's all I need to set one layer. So that's relatively simple. You've got a few options down at the bottom for how you would like to do this, and then you click do training. So this thinks for a little bit, and when it's done, you have an output. So I ran this earlier, and you can see that my result was, I could name it, so I called it PDCs versus not in a very Borat kind of manner of naming. You can see what we could potentially apply this trained neural network to. You can see the output of its training. Happily, it got very nice and accurate. On the training data, we actually are fairly happy that it wasn't perfect the whole way. The validation one was perfect. We weren't overfitting, perhaps. You can see that there were definitely some interesting epochs in there, and that the training loss got pretty rough. Interesting, the validation loss was already better. And then nicely, we can see the images that it said, okay, here's our example of our training images. Okay, so we brought some nice bright pink ones in. That looks very promising. All right, and at any point, you could click on a population and say, please find the PDCs in this one, and then just hit the do predictions button. Do prediction. So that would think for, probably like 15 minutes. And then we would show predictions, and it would show you some examples of, these are just basically some probability. So it's, just showing some simple count graphs, some neural network plots, and then it will actually then pop in some gates into your layout editor if you hit apply. All right, so that's the gist of how we anticipate the neural networks working. Again, they're working on Macs at the moment. We still have to figure out how to make the libraries work for PCs. We still have to figure out what the best neural networks are for this. We still have to benchmark which ones seem to work well in this data and which ones don't. But I just wanted to show you a little taste of where our thinking is going in terms of automating more of this process. Okay, we've got 5 minutes left. And so I would like to wrap up there and leave at least 5 minutes for questions. And I will pop the video back on so we look like people talking again, and check the chat box out, which appears like I am caught up at the moment. So if you've got a question for now, feel free to pop it into the chat box. I will remind you that my email address is john.flojo@bd.com. If something occurs to you in the future that you would like to send my way, happy to chat. Let me take a sip of tea and give people a chance to type. And while people are thinking, my screen is sharing, I think it's worth mentioning that our website is flowjo.com. And if you want to find recorded webinars at some point in the future, they are just at the bottom of the webinars list. Goodness, hopefully your internet is moving faster than mine. And hopefully that means you're still there. There we go. So learn webinars. Here's the one we're looking at. And then previously recorded webinars is here. and we keep about a 2-year backlog of webinars. All right, for the folks who are watching this recorded, I will assume you've already found this. Okay, the chat looks empty, the Q&A looks empty, and we're about at time, so I will just assume you're a happily satisfied audience and wrap up today's discussion. Thank you again for joining us. It's been my pleasure to show you this new technology.